Hello and welcome! Today, we will focus on LO4, understand the mode of action of enzymes. We'll explore how enzymes act as biological catalysts, speeding up reactions in our cells and allowing life to thrive at normal body temperatures. Correct. Enzymes are proteins with a specific three-dimensional shape. Their function is dependent on this precise structure. Each enzyme is specific to a particular reaction and acts on a specific molecule called a substrate. They remain unchanged after the reaction. Without enzymes, biochemical reactions would be too slow to support life. Every enzyme has a region called the active site. The active site has a specific shape that is complementary to the substrate. It forms temporary bonds with the substrate. When the substrate binds to the active site, an enzyme-substrate complex is formed. This process is crucial for the reaction to occur. This model explains enzyme specificity. The active site is rigid, and the substrate fits exactly into it, like a key fitting into a lock. Once bound, the reaction occurs, and products are released. The enzyme is then free to act again. This model highlights the precise match between enzyme and substrate. This model provides a more accurate explanation. The active site is flexible and changes shape slightly when the substrate binds. This improves the fit and helps the reaction occur. The induced fit model explains how enzymes stabilize the transition state and lower activation energy. It is widely accepted for its accuracy. Chemical reactions require activation energy to start. Enzymes lower this energy by bringing substrates close together and correctly orienting molecules. They also strain bonds in the substrate and provide a favorable microenvironment. By lowering activation energy, enzymes make reactions faster and more efficient. This process is vital for life. Temperature affects enzyme activity significantly. Increasing temperature raises kinetic energy and reaction rate until an optimum is reached. High temperatures can denature enzymes, changing their shape. In humans, the optimum temperature is around 37 degrees Celsius. Beyond this, enzyme activity declines sharply. Each enzyme has an optimum pH. Extreme pH levels can alter the active site and reduce enzyme activity. For example, pepsin works best in acidic conditions in the stomach. Outside of its optimal pH, an enzyme's efficiency decreases. Maintaining the right pH is crucial for enzyme function. Increasing substrate concentration increases reaction rate until enzymes become saturated. At saturation, the reaction rate reaches a maximum, known as Vmax. Beyond this point, adding more substrate does not increase the rate. Enzymes can only work as fast as their active sites can bind substrates. This is a key factor in enzyme kinetics. Enzymes are highly specific due to their unique amino acid sequence and precise three-dimensional structure. The chemical properties of the active site also contribute. For instance, amylase breaks down starch, while lactase breaks down lactose. They cannot substitute for each other. This specificity is essential for their biological roles. An investigation can explore how temperature affects enzyme activity. As temperature increases, enzyme activity rises to an optimum. Beyond this point, enzymes denature, reducing reaction rates. Such studies illustrate the balance required for optimal enzyme function. They highlight the importance of maintaining stable conditions. Similar investigations can be conducted for pH effects. Using buffers at different pH levels, enzyme activity can be monitored. Each enzyme has an optimum pH, like pepsin in acidic conditions. Extreme pH levels disrupt ionic bonds and change the active site shape. These studies emphasize the need for pH balance. Enzymes control all metabolic processes in living organisms. They work by binding to specific substrates at an active site, forming an enzyme-substrate complex. This lowers activation energy and releases products. Their activity depends on temperature, pH, and substrate concentration. Without enzymes, life-sustaining reactions would not occur efficiently. Enzymes play a crucial role in maintaining homeostasis. They ensure reactions proceed at rates necessary for life. Environmental conditions like temperature and pH must be optimal. Any deviation can lead to enzyme denaturation and loss of function. Homeostasis is vital for survival and enzyme efficiency. Enzymes are essential biological catalysts that facilitate life processes. They work by binding to substrates, forming complexes, and lowering activation energy. Their efficiency depends on optimal conditions like temperature and pH. Understanding enzymes is crucial for grasping how life functions at a molecular level. For further questions, please contact us on WhatsApp at 074-8436-1688. Thank you for listening. We'll see you at the next session.